Hello and welcome. The topic before us is the tricarboxylic acid cycle, also known as the TCA cycle. The TCA cycle is a central pathway of cellular respiration. As we start, let's take an overview of what the TCA cycle is. The TCA cycle is also known as the Krebs cycle or the citric acid cycle. In the TCA cycle, a series of chemical reactions occur. These reactions generate energy through oxidation of acetyl-CoA. The TCA cycle occurs in the mitochondrial matrix and produces a number of biomolecules. It's central to energy metabolism and biosynthesis of biomolecules. What are the key functions of the TCA cycle? One is energy production. It generates energy in the form of ATP, NADH, and FADH2. Two is biosynthesis. The TCA cycle provides intermediates for amino acids, lipids, and nucleotides biosynthesis. The third function is carbon metabolism. The TCA cycle converts acetyl-CoA into carbon dioxide. There are several steps involved in the TCA cycle. The first step is the combination of acetyl-CoA with oxaloacetate to form citrate. Now, when citrate is formed, it undergoes isomerization to form isocitrate, which is oxidized to alpha ketoglutarate, producing a molecule of NADH. Alpha ketoglutarate is converted to succinyl-CoA and produces another molecule of NADH as well as carbon dioxide. Succinyl-CoA forms succinate and produces GTP or ATP. Succinate is oxidized to fumarate and produces FADH2. Fumarate is hydrated to form malate, while malate is oxidized to oxaloacetate, and another molecule of NADH is produced. Now, if you look at the steps, you would understand and see that three molecules of NADH are produced, a molecule of FADH2 is produced, and a molecule of ATP or GTP is produced. Now, the diagram here shows the tricarboxylic acid cycle, known as the TCA cycle. And pyruvate produced from the glycolytic pathway produces acetyl-CoA, which condenses with oxaloacetate to form citrate. Now, citrate undergoes isomerization, like we have seen in the step in the previous slide. Citrate undergoes isomerization to form isocitrate. And isocitrate undergoes oxidation to form alpha ketoglutarate and NADH produced with a molecule of carbon dioxide released. Alpha ketoglutarate produces succinyl-CoA. There's also the release of carbon dioxide and the production of NADH. Succinyl-CoA converts to succinate and GTP is produced, which is either GTP or ATP. This is adenosine triphosphate produced from GTP. Now, succinate is oxidized. It produces FADH2 molecule. It produces fumarate. And fumarate is hydrated to form malate. And malate is converted back to oxaloacetate with the production of NADH. And oxaloacetate condenses again with another molecule of acetyl-CoA produced by the decarboxylation of pyruvate. And then the cycle continues to produce energy. What is the energy yield of the TCA cycle? We want to calculate the energy yield of the TCA cycle. So per ton of acetyl-CoA that condenses with oxaloacetate, we have 3 molecules of NADH produced. Each molecule of NADH will give 2.5 molecules of ATP via oxidative phosphorylation. And so 3 molecules of NADH will produce 7.5 molecules of ATP via oxidative phosphorylation. 1 molecule of FADH2 is produced, and for a molecule of FADH2 produced in the TCA cycle and undergoes oxidative phosphorylation, it will produce 1.5 molecules of ATP. 1 GTP produced is equivalent to 1 molecule of ATP. So the total yield of ATP from a molecule of acetyl-CoA that is completely oxidized through the TCA cycle is 10 molecules of ATP. 10 molecules of ATP are produced per molecule of acetyl-CoA that is completely oxidized through the TCA cycle. Now we want to look at the regulation of the TCA cycle. There are key enzymes that help in the regulation of the TCA cycle. The first is citrate synthase. Citrate synthase, which is inhibited by high levels of ATP and NADH. Isocitrate dehydrogenase is also activated by high levels, of adenosine diphosphate and is inhibited by high levels of adenosine triphosphate and NADH. Alpha ketoglutarate dehydrogenase inhibits or is inhibited by NADH and succinyl-CoA. Then energy demand. The cycle activity increases when ATP levels are low. The activity of the cycle increases so that more ATP can be produced for the cell to carry out its function. What are the clinical significance of the TCA cycle? Mitochondrial disorders. Mutations affecting enzymes of the TCA cycle can impair energy metabolism. Cancer. Tumor cells often exhibit altered TCA cycle activity. Drug targets. Certain antibiotics and anti-cancer drugs target enzymes of the TCA cycle. In summary, the TCA cycle is central to cellular energy metabolism. It produces key metabolites for energy in the form of ATP, NADH, and FADH2. The TCA cycle plays a vital role in biosynthesis and regulation of metabolic pathways. Thank you very much for good listening. Until we see another time, we'll call it a day. Thank you.